Hello, and welcome to this lecture on the use of CRISPR in human gene therapy. So to start out with, we've already talked about a variety of different genetically modified organisms within this course. And just as a quick review, within the genetic modified organisms, what you're looking at is where scientists or possibly corporations have taken and inserted in different genes into an organism, into the germline of the organism. So we get a transgenic line where all of the offspring of this kind of new created variation of the species are going to be genetically modified in order to express a specific type or category of proteins. An example of this is the Glofish. These were originally done as zebrafish to express fluorescent proteins as a marker for looking at the levels of pollution within waterways. The FDA, the Food and Drug Administration, ruled that because it wasn't a food source, they didn't pose a greater risk to the environment than the normal zebrafish. Still, these types of fish are illegal in California, Canada, European Union, and Australia. What's interesting is that over the last few years, in addition to the fluorescent zebrafish, the company that's doing this Glofish has created a series of, looks like another 6 or 7 different species of fish that you can now get at aquarium stores that will glow. So you can put them in a black light aquarium and they're going to fluoresce. But the focus on today's lecture is to look at concepts related to gene therapy and an effort to try to correct these genetic disorders to minimize the impact of these inherited diseases. And so, according to the NIH, it's estimated that about 2% of newborns within the United States are going to be born with a genetic disorder. And their reference section, the genetics home reference, indicates there's probably between 2,000 and 3,000 inherited diseases. Some of these, are more complicated, and there's cross traits or cross characteristics caused by multiple genetic disorders. Sometimes individuals have one, sometimes individuals have multiples of these genetic disorders. Some examples of these, which we'll talk about a little bit within today's lecture, are going to be things like cystic fibrosis, which affects about 30,000 Americans, and adenosine deaminase deficiency, ADA, which affects about 40 to 50 people worldwide. So again, you can see the range of individuals that are affected by specific types of genetic disorders. Now with gene therapy, the idea is that we're going to use some of the advances that we've developed in genetic recombination to take these recombinant DNA technology and our understanding of kind of the correct gene to try to treat the human disease by fixing the defective gene. And so, as illustrated on the image on the slide, the idea is that you take some type of therapeutic gene, you put it in a vector of some type that is then going to get into the individual cells, and in doing so, normally just place in kind of a corrected copy for that defective gene. Very rarely in the past, there would be an effort to actually go in and directly swap the replacement or corrected, what's illustrated here is the therapeutic gene, replace that therapeutic DNA with the defective DNA within the damaged cells, or the damaged genome. In the past, this really wasn't possible, but this may change in the future with some of the techniques associated with CRISPR. Now, when we're taking a look at gene therapy, and most commonly when we talk about gene therapy, we're talking about somatic cell gene therapy, so treatment of kind of normal body cells, and so somatic cells, body cells, as opposed to germline. The germline cells are going to be the egg and the sperm, and potentially if we, alter the genes within the egg and the sperm cells, potentially we could be altering future generations as well. And so up until this point, predominantly what we've been looking at is going to be somatic or body cell gene therapy. And what we've learned over the last probably 20 to 30 years where we've made efforts at using gene therapy is that we have to get the DNA into the cells. And so we have to use an appropriate vector. And in many cases, we're going to be using kind of a variation of primarily a cold virus. That cold virus then is going to be able to, infect the cells, and it does what viruses do. The viruses, when they infect the cell, are going to inject their genetic material into the cells. And so the hope is that when we use a virus as a vector, it's going to insert in kind of our correct DNA into the cells. Once the DNA is within the cells, it needs to be expressed. And again, oftentimes when we use a virus, it's got a very strong promoter associated with it. And so once it's in the cells, regardless of where it is within the genome, it's going to be expressed fairly well. However, what we've learned, again, over 30 years of trying these techniques is that mainly what we're going to be targeting is normally rapidly dividing adult cells in areas of the body where the DNA can get into the cells. As the cells divide, they're going to copy that corrected DNA, and we're going to get lots of copies of those cells that are going to be expressing the correct form of the gene, ideally correct protein, for whatever it is that we're attempting to treat. But the problem is that in many cases, if we're looking at rapidly dividing adult cells, these are usually in areas where we're going to have rapid turnover of cells. And so, we're going to look at an example in a couple slides where we've got cells within the lining of the respiratory system. They're continually going through a process of renewal and replacement. And so while we may be able to go in and use a cold virus to transfect these cells to get the DNA into them, those cells are going to be turned over a period of time. And so we have to repeat that treatment. There's also evidence that at least some of these cells may stop expressing that new DNA. And therefore, once they stop expressing the DNA, we have to go through and do another round of gene therapy to kind of insert the DNA into a new set of cells, hopefully to get those cells to express the DNA and treat the condition that we're trying to correct for. There's also pretty good evidence that the immune system is involved when we're looking at gene therapy. If we think about what's going on within gene therapy is that we're putting in a correct form of a gene, and that gene in many cases is going to code for something that's going to be produced by the cell, protein that's going to be produced by the cell. And if it's different from what was normally found within the body, normally found within the cells, often the immune system is going to recognize those cells that have been viral infected and have the correct DNA inserted into them as being potentially foreign or at least potentially pathogenic, and are going to attack the cells, kill them off, so that we've got to do another round of gene therapy to infect a new set of cells. The problem is that once we've got an immune response, each round of gene therapy, if we do it in a traditional sense, is likely to elicit this immune response, and we get a quicker and quicker kind of elimination of the cells that we've tried to alter by putting the new DNA in. Now, one of the earliest examples of gene therapy was an effort to treat cystic fibrosis. Now, cystic fibrosis is an inherited disease. It's associated with a single gene defect. And the gene associated with cystic fibrosis was identified in 1989. The gene with the impact on the majority of cases of cystic fibrosis is a cystic fibrosis transmembrane regulator gene. It basically codes for a protein that is found within the epithelial lining cells within the airway. And these cells are normally going to be pumping chloride across their membrane, putting chloride into the airway space here. And what happens is that water is going to follow that chloride, attempt to, kind of dilute it as it's in there. And in doing so, what it does is that water accumulates and it's going to kind of dilute the mucus that is normally found within the respiratory system. In individuals that have cystic fibrosis, they have damage to the cystic fibrosis transmembrane regulator protein. So they don't pump chloride across the surface. Because they're not pumping chloride across the surface, water isn't following behind. And so that mucus isn't diluted and kind of washed away. That mucus is going to start to build up. As that mucus builds up, it, potentially damages the airways, potentially blocks the airways. It also serves as a location where, bacteria can kind of get seeded into the mucus. And so you run the risk of a bacterial infection, damaging of the wall of the blood vessels, or not the blood vessels, the airway passages, because of the buildup of the bacteria and the mucus. Often get a little bit of blood coming in, again, because of the damage there. And so you really disrupt the airway passages. The idea with cystic fibrosis is that we're going to take that gene for the cystic fibrosis transmembrane regulator, for the protein that pumps the chloride across the membrane. We're going to put it into a vector, most commonly a common cold vector, that can be inhaled by the person with cystic fibrosis. That's going to get these viral particles down deep within the lungs, where they're going to infect the cells. But instead of infecting the cells with the common cold, they're going to infect the cells with the proper gene for cystic fibrosis transmembrane regulator protein. And in doing so, by getting these cells to express this transmembrane regulator protein, that protein is going to be present along the surface. We're going to pump chloride across it, and we're going to get dilution of the mucus within the air passageways. Again, these cells often go through rapid turnover, so we need to do another round of gene therapy, as well as the expression of this now regulator protein on the surface of these cells could elicit an immune system response and result in the immune system attacking these cells. Now, it's been commonly used with these cold viruses as a treatment method. More recently, we've seen scientists and researchers attempting other delivery methods. One example of this is in 2015, a group of scientists in Britain looked at using the viruses within fat droplets. And so the idea is that they would end up with kind of a better delivery method of the viruses. And so, we've got a patient here on the right-hand side of the slide using kind of a traditional nebulizer that a lot of patients with respiratory issues use to get medicines down kind of deep within their lungs, often used for like asthma medications and things like that. But they get better delivery of these fat droplets carrying the viruses and transfecting the cells within the respiratory epithelium. And they've actually got to phase 2B clinical trials showing that repeated doses were very effective in treating cystic fibrosis. And it seemed to be better than just using the viral particles alone. With many of these techniques, what we see is that not all patients are going to respond similarly or equally to the treatment. And so there's still a lot that we need to learn about these concepts related to gene therapy. Another example, and probably the most successful example of human gene therapy at this point, again, still somatic cell gene therapy, is a treatment for a severe immune deficiency disease. This is the ADA, SID, the adenosine deaminase, severe combined immunodeficiency. What you see with these patients, often very, very young patients, is that they have a defect in the gene for adenosine deaminase. And because of that, they have a reduced number of lymphocytes and an impaired immune system. And so they're going to be very susceptible to infections. So much so that these patients are often referred to as having the bubble baby syndrome, because they have to be completely isolated from others, or they're at a huge risk for receiving potential infections. And because they can't mount an immune response, those infections that would normally be fought off by the immune system can be very devastating and potentially lethal to these patients. And so the idea with this is, again, they take the bone marrow cells out of one of these patients. They alter them by transfecting in the correct gene for ADA into these cells. The cells then are going to start to express the new adenosine deaminase. They're going to be cultured, they're going to be identified, and then once they've got a population of these cells going, they're going to inject these cells back into the bone marrow and then hopefully kind of replenish those hematopoietic cells, replenish the cells that are producing the lymphocytes, and the lymphocytes now are going to be functioning properly. Most recently, the Food and Drug Administration within the US approved another method of gene therapy for spinal muscular atrophy. This is, again, a very leading cause of infant mortality. In this case, we've got damage to spinal motor neurons, resulting in atrophy or weakness. And they've been able to, again, use these cold vectors. The adeno is a cold virus. So adeno-associated viral vectors to carry in the correct gene. They've been able to see sparing or growth of these spinal muscular neurons, and it seems to have a good effect in preventing kind of the wasting of the muscle and the weakness that's observed in these patients. This is an example of the FDA kind of rapid approval for something. They've been able to fast track this treatment, giving it a priority review and an orphan drug designation, and so they're able to get this from kind of design through the clinical trials relatively quickly and allow it to be treating these patients very, very quickly. If we take a look at gene therapy over the last few years, this is from 2017, the majority of the gene therapy clinical trials have been focused in on cancer diseases and monogenic diseases, diseases with a single gene affecting them. So there are a number of different types of conditions that are being looked at, and this continues to grow with each year. Now, most of the gene therapy up to this point has been a somatic gene therapy. And again, somatic body cells, you're going to be targeting the cells, generally a small subset of cells, whether they're treated within the individual or isolated from the individual, altered in a laboratory setting and then put back into the individual. It works, but in general, it requires multiple treatments, and it doesn't result in stopping the transmission of the disease to future generations. And so, kind of at least from a theoretical standpoint, one of the goals is to look at germline mutations and germline therapy. Now, with the concept of this, instead of putting our corrected DNA into somatic or body cells, which aren't going to be transmitted to the next generation, by putting it into germline cells, you're going to end up with every cell within that new offspring, every cell within the individual, the baby that's being formed by this, are going to have the transplanted DNA as opposed to a few subset of cells in somatic. And we've in essence kind of altered the germline. We've altered the genetics of the information that will be passed from generation to generation. And so potentially it could cure an inherited disease, but there's a lot that needs to be understood about this because we're changing the genome and having an effect on all of the offspring, all the progeny kind of going forward from an individual kind of in, basically indefinite, all the offsprings, all future generations of the offspring. So the concept with germline gene therapy is that we're going to take the corrected DNA, we're going to insert it into the equivalent of a fertilized egg. It's going to be incorporated by the DNA within the fertilized egg, and then as that egg develops, all of the cells are going to have that corrected DNA. And so while we're talking about this as germline gene therapy within a human kind of clinical setting, what we're talking about is basically the same procedures as if we were creating a transgenic organism, a genetically modified organism, because in reality, that is what we're doing. We're genetically modifying the information that's going to get into all of the cells of that new individual, including the germline cells. And so potentially the egg cells or the sperm cells are going to be produced so that that modification is going to be passed on to future generations. When we're looking at transgenic organisms, that's our goal, because we want to do that genetic modification and then have all of the progeny that are generated from that kind of founder, that individual that has the corrected DNA, or at least the inserted DNA into it. We want all of the progeny to express it, so we don't have to go through the recombination and the DNA programming over again. Now, most recently, within the last probably 5 years or so, maybe 5 to 10 years, we've had this really incredible kind of advancement in the ability to edit DNA with greater accuracy. And this is the CRISPR technique. The CRISPR, which I think most of the sections have talked about, at least at some level within this course, is a clustered, regularly interspersed short palindromic repeat. It basically kind of allows kind of a targeting of specific regions of the DNA. It's associated with a bacterial enzyme, kind of like the restriction enzymes that we've used previously within molecular biology techniques. But the combination of the Cas with the CRISPR system allows for specific targeting of a unique region, a distinct region, a targeted region of the DNA molecule. Now, the CRISPR-Cas9 method was developed by biochemists Emmanuel Charpentier, Jennifer Doudna, and Feng Zhang. And so they're getting a lot of attention very recently because of all of the advances that are now possible with this technique. And just, kind of a quick commercial here and aside, this is kind of Nobel Prize level work. And so very seldom do we have the opportunity to really see kind of monumental kind of scientific shifts occurring. This is the type of thing that could really change the area of kind of DNA manipulation, genetic recombination, for a whole variety of both altering DNA, kind of improving DNA, correcting, defects, putting in, transgenic organisms, genes that we want to be expressed. This allows for a much greater level of targeting than was previously possible, and minimizes potentially a lot of the risks of not knowing where we're going to be inserting in our DNA, and potentially causing kind of future problems by correcting kind of an initial kind of defect within the DNA. So again, I think most of the classes have talked about this a little bit before, but basically how CRISPR works is that we're going to have a protein complex, this Cas9 complex here, which is going to have the ability to cut the DNA. It's going to interact with a piece of guide DNA. So that piece of guide DNA allows us to go through and sequence our targeted region of the DNA molecule, create our guide DNA to be a complement to that. So it's going to match up very specifically to the area that we want to cut. And so our guide DNA is going to allow us to kind of go in, recognize a specific region of the DNA, and then the Cas9 protein, as an example of another restriction enzyme, is going to go through and cleave that DNA, cut the DNA at a specific point. So we're going to cut that DNA, and then with the process that's been developed with CRISPR-Cas9, we can then, again, use the normal kind of correction mechanism, the repair mechanism within the DNA to insert in kind of our correct gene or corrected kind of sequence or inserted sequence that we're interested in, and place that DNA in a specifically targeted point. And so this, again, allows us to go in and specifically target a unique region of the DNA to either insert in a corrected gene or a new gene in a way that wasn't possible, even 5 or 10 years ago. Now, if we take a look at this technique, it has a lot of applications, and one of those applications is that we can go in and specifically target identified regions of our DNA molecule, so identified regions of cells that we're interested in treating. In 2018, a Chinese scientist named Jiankui reported the birth of genome-edited twin girls using the CRISPR system. And so basically, he was working with subjects in which the father was HIV positive, but they wanted to have offspring. They wanted to, have, children, and minimize the risk that the HIV would be transmitted from the parents to the offspring. And so what Jiankui did was he basically used the CRISPR system to go in and induce a mutation to disable the genetic pathway that HIV uses to infect cells. And he basically targeted the Chemokine receptor 5 protein receptor, which is found on white blood cells, and it's used by HIV to infect white blood cells and basically kind of attack the immune system as well as cause the HIV AIDS disease. Now, he reported this at the end of 2018. It resulted in almost immediate condemnation for the act and a call for international regulations on what should and shouldn't be allowed to be done when you go through and potentially are editing the genome, the germline editing of eggs and sperm cells, and potentially kind of altering future generations. Now, for Jiankui, this didn't end up very well. There was a number of investigations over the last year or so looking into his work. What it was determined is that there were at least 3 areas in which there were ethical concerns raised about this, well beyond kind of just going through and doing the germline editing. He's gone through, he's altered the genome. We've had the children being born with that, but we don't know the long-term effects of the genetic changes that he's induced. Did he cause other problems that are now going to be transmitted? So created a new inherited disease that could be found within these individuals, these children that were born, or their progeny at some point in the future. There's also, it's not clear what going through and inducing this mutation is going to do and make them susceptible for going forward. There were also issues in regards to safety testing. As I said on the previous slide, he was recruiting couples with an HIV-infected father. He basically kind of altered some of the records, altered some of the testing of the samples so that it wasn't clear that one of the subjects was HIV infected, and potentially, put people in his laboratory at risk associated with this. As well as he didn't follow standard procedures for recruiting patients and subjects and kind of protecting their interest in this. One of the things that came out was that once they started in this kind of fertility, it's almost like a variation of in vitro fertilization. It's in vitro fertilization with the altered kind of DNA kind of going into it, which is a very expensive process. He basically told the patients that if they dropped out of the study or kind of withdrew their consent, they would have to pay for all of the treatments that it had received at that point. And because it's very expensive, this was seen as being coercive, because you're basically forcing people to kind of stay in the study, even though they may voluntarily want to get out of it, but they don't have the means to, because they don't have the financial means to be able to pay, in essence, to be able to get out of that study. So he ended up being sentenced to 3 years in jail at the end of December 2019 for what was determined in China to be illegal medical practices. So you would think that this would be kind of the end of the story for this, but there are other individuals that are still attempting to pursue these types of strategies of going through and doing gene therapy. One example of this is a scientist, a biologist in Russia that has been using the CRISPR techniques, basically the same type of techniques, to go through and edit genes that have been donated by individuals. Some of the things that he's been working on are allowing HIV positive mothers that didn't respond to medication to have an HIV negative baby. So basically do a correction similar to what Jiankui did. He's also looking at kind of correcting kind of the genetic disorder associated with a genetic hearing loss that would potentially allow a deaf couple to have a baby without hearing loss. And as recently as October 2019, he was continuing his work in the lab, continuing to work on these eggs and manipulating them. But he wasn't to the point of implanting these eggs through the process of in vitro fertilization, selection, and implantation into a surrogate mother, or into the actual donor mother. He's not quite to that point yet, and he has said that he's going to follow the internationally agreed upon regulations. But even though there's lots of controversy associated with it, lots of discussions going on, we still don't have clear rules on what should be allowed in relationship to germline editing and the application potentially of CRISPR. So if we look at what Jiankui, get the name right, Jiankui did, the Chinese scientist that was actually altering the genome so that the children of the HIV infected father aren't going to be at risk of getting HIV, is he basically looked at targeting the CCR5, the Chemokine receptor 5 gene that's found on white blood cells. Normally, these cells, as illustrated on the diagram to the right, are going to have these chemokine receptors on the surface, and this is used by the HIV viruses as an anchorage point that bind to it and then actually get incorporated into the cells and infect the cells. By inducing the mutation in the CCR5, you basically result in kind of the truncating of the protein, and the protein's not expressed along the surface of the cells. And so the idea being that without that protein on the surface of the cells, HIV can't come into contact with it, can't dock with it, and can't get into the cell and infect the cell. And so kind of at least at kind of the biological kind of question, how does it work? It makes sense. But we don't know the long-term impact of having that deletion, the loss of that CCR5 protein on the white blood cells of the individual. Now, this is roughly similar to a CCR5 delta 32 deletion that is found normally within the population. It's found in about 10% of people with Northern European descent. It's a 32 base pair deletion mutation, which results in kind of a shortening of the protein. It's a termination of the protein before it gets very far, so they don't have that protein along their surface. And as scientists have studied CCR5 and this deletion, what they've been able to determine is that there's some advantages associated with it, that it protects against the bubonic plague, protects against HIV, which we kind of known from this slide here, and potentially protects against smallpox, again, probably through a similar mechanism. Still, we don't know everything that the CCR5 protein is doing within these cells. And so it becomes kind of a serious ethical issue, a serious biological question. Do we understand enough about the system to be able to go through and start manipulating it, kind of reversing, at least hundreds of thousands, if not millions of years of evolution to alter the DNA in some way based on our current interpretation of what's going on? So an example of that is if we take a look at sickle cell anemia. I think a lot of you have heard of sickle cell anemia before. Basically, we're looking at a damaged gene within the hemoglobin. It's a single amino acid chain, single amino acid change within the polypeptide, the protein for hemoglobin, which impairs the ability of the hemoglobin impairs the ability of the blood cells to carry oxygen. So under normal circumstances, an individual with sickle cell anemia are going to have kind of normal looking red blood cells, but in areas where there's less oxygen tension, at higher elevations, or as a result of kind of increased exercise or some other things that can kind of impact kind of the oxygen levels that are available. These cells are going to start to have a sickled appearance. They're going to start to have this irregular kind of clumping, almost like appearance to the red blood cell, the sickle cell appearance. And this can be very harmful to the individuals that have sickle cell anemia. It can result in blood clots. It can result in very painful situations, again, because of the impaired blood flow that's going on with the lower oxygen tension and these sickled cells. And so this is kind of a prime target for going through and correcting it. It's a gene that we've understood for a long period of time. It's a single amino acid chain. So in theory, we could go in with CRISPR and target this very easily. The problem is, again, we don't know everything that sickle cell anemia may be doing because it's still present within the population. It's still a gene that we see. It hasn't been kind of evolutionarily lost over time. So there's got to be at least some advantage to it. And more recent studies have shown that individuals that have one copy, so they'd be heterozygous for the sickle cell anemia gene, the defective gene, have a resistance to malaria. And so in areas that are tropical or subtropical with a high incidence of malaria, they have an advantage by having the sickle cell anemia rather than having, two copies of the corrected gene. If they have two copies of the corrected gene for hemoglobin, they're going to have normal functioning red blood cells. They're not going to have the traits associated with sickle cell anemia, but they're also going to be highly susceptible to malaria. So when we're thinking about going through and altering the genetics in some way, again, to take a look at this from an ethical perspective and from a somewhat historical perspective, what we're talking about at some level is almost a form of eugenics. Now, eugenics is an effort to improve the genetic characteristics of humans. This was started in about the 1800s, probably around the 1880s, and has continued since then. Eugenics was a main kind of driving force within Nazi Germany to establish a master race. And so they did, selective breeding to, promote the Germanic stock with the kind of ideal kind of German characteristics that they wanted to see going forward, as well as euthanasia. They went through and killed many people that didn't fit into their category of kind of the ideal situation. And so we can see, at an extreme, how bad that is, kind of just, it's beyond bad. I can't even think of a better word, that goes along, that conveys how awful that approach was. But having said that, it wasn't, the eugenics movement wasn't only found within Nazi Germany. It was very common around the world and even beyond World War II. Examples of that within the United States were the forced sterilization of patients in mental hospitals and convicted criminals that continued into the 1960s around the world, as well as the US. Even more recently in the 80s, there was an effort to kind of encourage sterilization of individuals within the criminal justice system to kind of minimize their potential for having offspring. And so again, this is something that's going on and continuing at varying levels within society, even if it's not recognized as being a true form of eugenics, or at least with the term eugenics associated with it. And so anytime we're talking about going through and altering the genome, we need to keep that in mind. It may make sense at a certain level to correct a genetic disease, but once we have that technique, once we have the technology, once we have the ability to do this, you know, it becomes kind of questionable. You know, do you correct disease? You know, where do you stop? Where is it permissible? Where isn't it permissible? And so some questions to consider after this kind of series of lectures, this lecture at least, when we're taking a look at, you know, potentially germline therapy. Should emerging recombinant DNA techniques like CRISPR-Cas9 be used to alter the genome of individuals with genetic disorders? You know, should we use this for, you know, somatic gene therapy? Should we use this for germline gene therapy? Should it only be permissible to treat lethal diseases to prevent the death of an individual? Or could we use this to treat genetic conditions, you know, conditions that may be, you know, difficult for the patient, maybe, you know, painful, or have disabilities or impairments associated with it? Should we go through and treat those? If we're going to treat those, is it permissible to use these techniques for genetic enhancement or potential customization of traits going forward? You know, so the concept of designer babies that were talked about, you know, in the late 80s and 90s, and, you know, never came about, at least to this point. But we're developing the technology where it then becomes a lot more feasible to actually pursue that, as we've got, you know, greater sequences of the DNA. We have a better understanding about the DNA, and we've got a better kind of tool within CRISPR-Cas9 to be able to go in and alter the DNA based on kind of our current understanding of what's going on. So just a few references associated with this, and again, I'll post these on Canvas site. So hopefully this series of lectures on research ethics are interesting, kind of generate some thoughts to think about. You know, especially when we're developing new techniques and new procedures, we're expanding our understanding of science, and we have to think about what we're going to be using that science for. So thank you, and have a great day.